Good morning, good afternoon, good evening, ladies and gentlemen. You are welcome to my YouTube channel. Today, we are going to be discussing about introduction to nucleic acids biochemistry. In this lecture video, you are expected to learn nucleic acid, nucleotides, and types of nucleotides. And at the end of this lecture, you should be able to describe the chemical components of nucleic acid, the formation of nucleosides, formation of nucleotides, and you should be able to describe DNA and RNA. So what is a nucleic acid, or what are nucleic acids generally? Nucleic acids are polymers of nucleotides. That is, nucleotides are the monomeric unit of nucleic acids. So nucleic acids, they are polymers of nucleotides, nucleotide being the monomeric or the building block of nucleic acid. So what are these nucleotides? Each nucleotide has three components. Each nucleotide has three components. A 5-carbon monosaccharide, which is a sugar, 5-carbon sugar, pentose sugar, which can be either deoxyribose if it is in DNA, or ribose if it is in RNA. We are going to use these two pictures of deoxyribose and ribose to explain the differences between the two. And apart from the 5-carbon monosaccharides, the second thing a nucleotide has is a nitrogen-containing cyclic compound, which is a nitrogenous base, which can either be purine or pyrimidine. We are going to see in the subsequent slides. And also, the last component of the nucleotide is a phosphate group. So each nucleotide has 5-carbon monosaccharide, 5-carbon pentose sugar, nitrogen base, and a phosphate group. So what is the difference between ribose and deoxyribose? We said deoxyribose is found in DNA and ribose is found in RNA. If you look at the deoxyribose here, if you start counting from this first carbon, carbon 1, carbon 2, in carbon 2, you pay attention to carbon 2. It has, because it's 5-carbon sugar, carbon 1, carbon 2, carbon 3, carbon 4, and carbon 5. So in carbon 2, you look at the H here, look at it. Carbon 2 has H in the deoxyribose, while in ribose, carbon 2 has OH. So deoxyribose in DNA, deoxyribonucleic acid, and ribonucleic acid has ribose sugar because it has OH at position 2. So take note of this difference. Now, so this is a typical structure or typical representation of a nucleotide. We said it has how many components? Can you remember? We just said it now, 3. It has a pentose sugar, right? It has a nitrogenous base, and it has a phosphate group. So you have 1, 2, 3 components. This makes up a nucleotide, okay? So let's proceed. Now, what are these nitrogenous bases? These nitrogenous bases are divided into 2. We have the purine bases, which contains 2 rings. Look at them. We have adenine, which is represented as A, and guanine, which is represented as G. These letters A, G, C, U, T are the ones you will be encountering in the nucleic acids, because it's going to be too ambiguous, too cumbersome to be writing the full names. So A is for adenine, G is for guanine. So adenine and guanine are they have 2 rings, and they are members of purine bases. Now, the second nitrogenous bases group pyrimidine bases, which consists or contains only one ring. You can see we have cytosine represented as C, uracil represented as U, and thiamine represented represented as what? T. So they have what? One ring. Okay. So adenine, guanine, and cytosine, they are both common to DNA and RNA, while uracil is only found in RNA and thiamine is only found in DNA. Okay, so in RNA, the thiamine that is found in DNA is being substituted with uracil. So instead of T, thiamine, RNA has uracil, but they have this cytosine, adenine, guanine, they have them in common. The only difference is that in RNA, instead of thiamine, you have what? Uracil. While in DNA, instead of uracil, you have what? Thiamine. Please take note of that. So what is this phosphate group? We said this phosphate group, is part of the what? Three components of that nucleotide. So it is very important component of nucleic acids. It serves as the link between two sugar molecules or nucleotides in polynucleotides. When we say polynucleotides, they are what? Polymer of nucleotide, is nucleic acid. That's what we are referring to as. So it serves as link between two sugar molecules or nucleotides. So you take note that this phosphate group is important in nucleic acid and it serves as link between two molecules of what? Nucleotides in polynucleotides. So later on, we are in subsequent slide, we will see how this phosphate group is used to link between sugar molecules, okay? So the phosphate group is a strong acid in solution. This explains why DNA and RNA are called what? Acids. It's a strong acid in solution. So that is why DNA and RNA are called acids. It is responsible for the negative charges that are found in DNA and RNA at physiological pH. So these are the 3 or 4 important notes that you take into consideration when you are talking about phosphate group in nucleic acid. So you remember we talk about nucleotides, right? So now what are nucleosides? Nucleotides, we explained that nucleotides have 3 components. They have phosphate group, they have pentose sugar, and they have what? Nitrogenous base. So nucleotides, the only difference between nucleosides and nucleotides is that nucleosides do not have a phosphate group. So nucleosides have ribose or deoxyribose sugar plus the nitrogenous base. So they don't have the phosphate group. So if you have nucleoside and you remove the phosphate group, you have what we call the nucleoside. So the combination of ribose or deoxyribose and one of the 5 bases produces a nucleoside. They are linked via a covalent beta-N glycosidic bond. Take note, this is the type of bond that exists between them. They are linked via covalent beta-N glycosidic bond. So you can see we have here, there are some nucleosides that are found in RNA and there are nucleosides that are found in DNA. So this is uridine, okay? This is what? Uridine. You can see this is uridine, this is cytidine, this is adenosine, this is guanosine, okay? So this is from uracil. The nitrogenous base is what? Uracil without the phosphate group. So you have what? Uridine. The nitrogenous base here is what? Cytosine. Without the phosphate group, you have what? The cytidine. So also adenosine, guanosine. These they are found in RNA. While the nucleosides that are found in DNA, you have what? The oxythymidine, because the pentose sugar here, the OH is absent. You only have what? Hydrogen instead of what? OH. So that is why it is the oxyribose. You remember? So In DNA, you have what? The oxythymidine, the oxycytidine, the oxyadenosine, and the oxyguanosine. So nucleotides now. Nucleotides are phosphorylated nucleosides. That is a nucleoside plus phosphate group. You have what we call nucleotides, or a sugar plus base plus phosphate group. That is a nucleotide. Each nucleotide is a 5-monophosphate ester of a nucleoside. So it's a 5-monophosphate ester of a nucleoside. So the phosphate group is attached to the hydroxyl group of pentose sugar by an ester linkage, usually OH group on carbon 5, 3, or 2. So for in this case, you can see that the attachment of the phosphate group is at carbon 5. So this is the nitrogenous base, which can be either purine or pyrimidine. This is carbon 1, carbon 2, carbon 3, carbon 4. This is carbon 5. You have one phosphate. You can name it nucleosides. If you have from here, if you can see, this is, if you draw the line from here, you don't have the OH, you don't have the phosphate group. So it means you have only the pentose sugar and the base, which is a nucleoside. So a nucleoside monophosphate, it has one phosphate group. Nucleoside monophosphate, right? Nucleoside diphosphate, right? And nucleoside triphosphate, NMP, NDP, and NTP. So all these nucleoside monophosphate, nucleoside diphosphate, and nucleoside triphosphate, they are all nucleotides. Okay? Now, let's look at the types of nucleotides that we have. Number one, we have monophosphate nucleotides. These monophosphate nucleotides are nucleotides containing only one phosphate molecule. So you can have three examples here, A, B, C. You have AMP, which is adenosine monophosphate. Look at the structure here, adenosine monophosphate, guanosine monophosphate, B, and you have what? Uridine monophosphate. So these are the nucleotide that contain only one phosphate molecule, from adenine, guanine, and uracil. So you have uridine monophosphate, guanosine monophosphate, and adenosine monophosphate. Now, the second types of nucleotide, we have diphosphate and triphosphate nucleotide, which they contain 2 for diphosphate and 3 for triphosphate. They contain 3 molecules respectively. Diphosphate and triphosphate contain 2 and 3 molecules of phosphate respectively. Example of diphosphate includes example of diphosphate nucleotide includes ADP, which is adenosine diphosphate, GDP, guanosine diphosphate, UDP, uridine diphosphate, while ATP is adenosine triphosphate. This is the energy currency that we're talking about for living organisms. ATP, GTP, guanosine triphosphate, and UTP, uridine triphosphate, are examples of what? Triphosphate nucleotides. The third category are cyclic nucleotides. In cyclic nucleotides, the phosphate group or phosphate molecule are esterified to they esterifize 2 OH molecule on carbon 5 and carbon 3, or carbon 3 and carbon 2 in RNA. So you have small letter C that is usually added to denote the cyclic nucleotide, is the example 3', 5', small letter C, AMP. It means 3', 5' cyclic adenosine monophosphate. You can have 3', 5' cyclic GMP. It's called what? 3', 5' cyclic guanosine monophosphate. And this fourth category are called polynucleotides. These polynucleotides are the DNA and the RNA. That is the polymer of the nucleotides. So here, the 3' OH of the pentose of a mononucleotide esterifies the 5' phosphoryl group of the second mononucleotide to form dinucleotide. So the OH, 3' OH of the pentose of a mononucleotide esterifies the 5' phosphoryl group of the second mononucleotide to form a dinucleotide. So the bond between the two nucleotides in polynucleotide is known as the 3' 5' phosphodiester bond. The next nucleotide joins the existing polynucleotides through its free 3' OH group. Okay, so each end of a nucleotide polymer, which is polynucleotide, is distinct. One end has a free 5' phosphate, while the other end has a free 3' group. So for each of these polynucleotides, there is going to be a free 5' and phosphate group. In 5', the phosphate group is what? Is free. While in the 3', the OH group is free. So by convention, nucleotides or base sequence is written in what? 5', 3' direction. So polynucleotides are called directional molecules. So DNA and RNA are long linear polymers called nucleic acids that carry information in a form that can be passed from one generation to the next. So genetic information is stored in the sequence of bases along a nucleic acid chain. So let's look at this picture explaining how the nucleic acid looks like. This is DNA structure. If you look at it, it has this nitrogenous basis. You have the adenine, thymine, guanine, cytosine. So this is how it looks like. It has the base pairing. You have adenine pairing with thymine. Guanine is pairing with cytosine. That is how letters are pairing. Thymine with adenine, cytosine with, so A with T, T with A, right? So G with C, C with G, that's how they pair, okay? The bond between adenine and thymine is 2 hydrogen bonds, while the bond between guanine and cytosine is 3 hydrogen bonds. You should take note of that. So it has sugar phosphate backbone, and it has this groove, major and minor group, and you have the base pairs. So if you look at this, this is a more opening structure. You have this direction, which is, look at it, 5', 3' direction. You remember at the 5' you have what? 5' phosphate group, which is free for joining, right? For chemical bond formation. And at the 3', you have the OH, the 3' OH, right? Available for bond formation. So adenine bonds with thymine with 3 hydrogen bonds. You can see 1, 2, while guanine and cytosine bonds with 3 hydrogen bonds, 1, 2, 3, you can see. So that is how they are pairing. Is from 5', 3'. This is a single strand, while the other single strand, making it the double strand, is from, look at it, 5', 3'. So that is how they pair. Okay. So DNA is what? Double stranded. Okay, molecule. Now, what is the difference now? Let's look at the difference between now DNA and RNA. Okay, you have in DNA, you have what? Double-stranded sugar phosphate. This is double-stranded. Okay, and the pentose sugar in DNA is what? Deoxyribose. While in RNA, you have a single-stranded molecule. It's a single-stranded sugar phosphate, and the sugar, pentose sugar, is what? Is ribose. Double-stranded in DNA, single-stranded in RNA. Sugar in RNA is ribose, while the pentose sugar in DNA is deoxyribose. So what do they have in common? They have cytosine, guanine, and adenine in common, but in DNA, you have thymine being the fourth nitrogenous base, while in RNA, you have uracil replacing thymine. Okay, so these are the observable features between DNA and RNA. Thank you very much. This is the end of today's lecture. I hope you have enjoyed it, and you are free. Feel free, please, to ask questions in the comment section. If you don't understand anything, and please like and comment this video, and also don't forget to subscribe if you have not done that. Thank you very much. See you in the next class.